Okay, so welcome to this next video in which we are discussing synchronous and asynchronous neurotransmitter release. Okay, so in this next video I want to now turn my attention to asynchronous neurotransmitter release. Okay, so far what we've seen is that when an action potential arrives in the axon terminal, it's going to cause the release of all of the readily releasable vesicle pool via synchronous neurotransmitter release. Now, what's going to happen basically in asynchronous neurotransmitter release? Firstly, what we need to do is get more vesicles to this presynaptic membrane here. And I'm going to first talk about the mechanisms underlying the recruitment of the reserve vesicle pool. And then what I'm going to talk about is how even when the calcium's starting to go down, so the action potential happened a long time ago, what is going to happen is you're going to be bringing in these reserve vesicles, docking them with the membrane, and then what's going to happen is you're going to continue fusing these vesicles that you've just docked with the plasma membrane, but it doesn't happen through synaptotagmin. Synaptotagmin no longer functions because the calcium's gone too low for synaptotagmin to be acting. So there's another protein suspected to be involved in this. Okay, so we'll begin our discussion of that then. Oops, we'll need another piece of paper. Right. Okay, so firstly let's discuss how we recruit the reserve vesicle pool. But firstly, actually, we should discuss what the reserve vesicle pool is. So, if I draw an axon terminal here, okay, so here's an axon terminal. Okay, then I've told you that we have this readily releasable vesicle pool, which is this store of vesicles in the axon terminal which are already docked to the presynaptic membrane. So this here, this represents the readily releasable vesicle pool. Releasable vesicle pool. Okay, so now I want to tell you what the reserve vesicle pool is. Now we don't just have a single store of vesicles in the axon terminal. Instead, we have this other store, which is a bunch of vesicles held sort of like not on the front line, basically. So they're held back. And this store of them of vesicles that aren't docked to the presynaptic membrane but are held further back, basically. This is what's known as the reserve pool. Okay, right. So, let me now discuss with you how these reserve pool vesicles are held, basically. How are they stored? Well, basically, within the cytoplasm of the axon terminal, you will have a cytoskeletal structure, basically. So you'll have fibers that are holding the cell together that are in this cytoplasm of this axon terminal. Now, some of these cytoskeletal components will be what are known as actin filaments. Okay, so let me discuss with you what an actin filament is. So an actin filament is basically a strand which is made out of the protein actin. Now, actin itself is a very small globular protein. So this little dot that I've drawn here, and I'll colour it in green, this represents an actin monomer, okay? But what will happen is that actin monomers can polymerize together like so. So you can join actin monomer after actin monomer after actin monomer and create a growing strand like so. So let me colour these in green. So this basically is a growing actin strand which we're making by polymerizing actin monomers together to make this actin filament, as it's called. So one of these huge great strands of loads of actin monomers stuck together, this is known as an actin filament. And these strands that you can make from this, these are what we can use as a cytoskeletal component. So basically, this axon terminal, it will be full of these actin filaments. So let's just draw them intertwining everywhere. They are what holds the cell up. It's not the plasma membrane, basically. When you think about what keeps cells in their shape, it's not the plasma membrane. The plasma membrane is a goo, okay? The plasma membrane is completely moldable. Instead, inside the cell is a meshwork, a very dense meshwork of protein fibers which are holding the cell in that shape. They are holding the plasma membrane up. And actin is one of the key components of this. So you're going to have actin filaments all over the place, basically. So here are actin filaments sort of intertwining and meshing around the cell like so. Now, basically we can attach synaptic vesicles to these actin filaments. Let me draw a synaptic vesicle here. So this is a synaptic vesicle with neurotransmitter in it. So here's our vesicle with loads of neurotransmitter in here. Now, there are certain proteins which can attach both to the actin filament and to the phospholipids of the vesicle, basically, within which the neurotransmitter is stored. Now I'm going to draw this protein here as a circle, but don't worry, I'll colour it in a different colour so we can distinguish it from the actin monomers. So this purple circle here, this is supposed to represent a protein which has a binding site for phospholipids on one side, so it can bind to the lipid bilayer of the synaptic vesicle on one side, and the actin filament on the other. And these proteins are known as synapsins. Okay, so this is a synapsin protein. Now, so this is how we store synaptic vesicles further back in the cytoplasm of the cell. They're not just floating around freely, basically. They are attached to the actin cytoskeleton of the cell by these synapsin proteins. So if we want to recruit the synaptic vesicles in the reserve pool to the readily releasable vesicle pool, for instance, because we've just released all of these vesicles in the readily releasable vesicle pool, so we want to restock it, we need to liberate some of the vesicles in this reserve pool so that they can diffuse over to the presynaptic membrane and then get docked there to replace the ones that we've lost. Okay, so we now need to look at the pathway by which you liberate these reserve synaptic vesicles. Okay, right. So, basically what's going to happen is that the calcium that you let in when an action potential arrives, i.e. the calcium that comes through these N-type and PQ-type voltage-gated calcium channels, it's going to, as well as activating synaptotagmin, it's also going to bind to a protein known as calmodulin, and I'll discuss this in more detail in a moment. Calmodulin is then going to activate an enzyme known as calcium calmodulin dependent kinase 2, and calcium calmodulin dependent kinase 2 is going to phosphorylate these synapsin proteins so that the synapsin protein no longer binds to the actin filament and also no longer binds to the phospholipids of the phospholipid bilayer of this synaptic vesicle. So basically the two are going to just be let go, basically, so you're going to let the synaptic vesicle go free. Okay, so now let's discuss this pathway in a bit more detail. So, calmodulin then. Calmodulin is one of these proteins that everyone despises because they don't really know what it is. So I want to give you a discussion of what calmodulin is. So, calmodulin in cartoons is often shown like so. Okay, so it has these two lobes known as the N lobe here and the C lobe here. And each of these lobes has a calcium binding site. So these are, it has two calcium binding sites. So each lobe, the N lobe and the C lobe, they both have two calcium binding sites. So a calcium combined in here, a calcium combined in here, calcium combined in here, and a calcium combined in here. So overall 4 calcium ions combined to the calmodulin protein. Now, these calcium binding sites are what are known as EF hand domains. Now I'll explain to you what an EF hand domain is. So an EF hand domain is a structure, it's a proteinaergic structure which is capable of binding to calcium. So if I draw a line to represent a polypeptide, if this is a polypeptide, then the structure of an EF hand domain looks kind of like this. The polypeptide is going to go down, it's going to form a ring like so, and then it's going to come back up. So this line represents the polymer of amino acids. So you've got amino acid followed by amino acid followed by amino acid followed by amino acid, etc. It's a polypeptide, a polymer of amino acids. Okay, now it forms this loop down here, and the amino acids which make up this loop are going to all have acidic side chains. Now, why does that help you bind calcium? Well, let me show you why. By giving you an example of an amino acid that has a acidic side chain. So we'll use aspartic acid as our example of an amino acid with a acidic side chain. So if this is the core amino acid structure, here's the alpha carbon at the center with our carboxylic acid group and our amino group up here, then the R group of aspartic acid is that you have effectively an ethanoic acid molecule sticking off the side. Okay, so this is aspartic acid. So let me write its name here. So this is aspartic acid, and it's strictly aspartic acid rather than aspartate, and I'll explain to you what the difference between aspartic acid and aspartate is. Now, the single letter amino acid code for aspartic acid is D. Just a bit of off-the-side information. Why is this going to be helpful in coordinating calcium? Well, basically, the important thing about acids, the thing that makes them an acid, is that they are capable of donating a proton. Now, basically, this carboxylic acid group here on the R chain here is capable of giving up that proton between the oxygen and the hydrogen. And I want to just stress that you might ask, well, what about this carboxylic acid group here? Why isn't this capable of doing the same thing? Why are you only talking about that one? This carboxylic acid group will be involved in an amide link with another amino acid. So these ones are going to be involved in amide links. So ignore those two. They're not going to be in their structure that I've drawn here. This is the pure amino acid. Remember, we're talking about the amino acid where it's viewed as being a residue of a protein. I should have probably maybe made that more clear by saying instead of that hydrogen there, we've got a link with another amino acid. And instead of this hydroxyl group, you've got a link with another amino acid down here. Okay, so now it's the residue of a polypeptide. Okay, now, basically, this R group, though, will be intact once you've put it into a polypeptide. So, this hydroxyl group here, you can break the bond between the oxygen and the hydrogen. Now, if we draw this bond out in a bit more detail, here's this single bond here. The hydrogen has provided an electron, and the oxygen has provided an electron. Now, what's going to happen when you break this bond is it's going to be done completely unfairly. Both of the electrons are going to go to the oxygen, basically, and the hydrogen is going to leave with nothing. So what's going to happen is the oxygen is going to gain a negative charge, and the proton is going to go off with a positive charge. So what you're going to get is you're going to get this carboxylic acid group now with just an oxygen here with a negative charge, and then the proton has been donated away. So that's why this molecule is an acid, because it's capable of donating protons. Now, the molecule of aspartic acid, once it has donated its proton away, i.e. the molecule when it's got this group with the negative charge here on the oxygen, is no longer called aspartic acid. Instead, it's called the conjugate base of aspartic acid. And the conjugate base of aspartic acid has its own name. It's actually called aspartate. So that is the strict difference between aspartic acid and aspartate. Aspartate strictly means aspartic acid, once it has lost its proton, once it's donated its proton away, it means what's left after that donation, basically, of the proton. Okay, so the reason you usually refer to this as the conjugate base as well is because this group now is no longer an acid. If you look at the aspartate molecule, it's certainly not an acid anymore. It can't donate protons anymore. Instead, it's actually a base because it can receive a proton on this negatively charged oxygen. So aspartate is a base. Aspartic acid is the acid. So you'd call aspartate the conjugate base of aspartic acid, and you could also call aspartic acid the conjugate acid of aspartate. So there's that sort of conjugation between the two. Right, now the important thing is that if you put loads of acidic residues in this ring, some of them will not have given up their protons, and they'll be neutrally charged. but some of them will give up their protons, and they will gain a negative charge. When they're in their conjugate base form, they will have a negative charge. So the point is, by putting a lot of acidic residues in this loop, you actually get a lot of negatively charged residues. And that is useful for coordinating calcium, because remember, calcium is a divalent cation. It's got 2 positive charges. Okay, so that's what an EF hand domain is then. Now, EF hand domains rarely occur on their own in proteins. Instead, what you usually find is you find them in pairs. So you find a structure that looks like this, basically, with a ring like that, then a linker, and then another ring. Okay, and when you've got 2 EF hand domains next to each other like this is what's known as an EF dimer. Okay, and these EF dimers, they are more common than just a single EF hand domain on its own. So basically, what can happen is each of these rings can coordinate a calcium ion. So a calcium ion can bind there, and a calcium ion can bind there. So in an EF dimer, you have 2 calcium binding sites. And these EF dimers, these are what calmodulin proteins have, what each of the lobes of calmodulin has. It has an EF dimer, basically. So you have an EF dimer on the N lobe and an EF dimer on the C lobe, and that gives you overall 2 calcium binding sites on each lobe. Now, back to the discussion of calmodulin. When it doesn't have calcium bound to it, it's in this sort of hunched over structure like so. And this structure is known as apocalmodulin, okay? And the shorthand for that is to call it apo C lowercase a capital M, like that. So this is the shorthand for calmodulin. A capital C, lowercase m, uppercase M. So CAM, basically, apocam. Now, when calcium binds to these calcium binding domains, when you get 4 calcium ions binding, 1 in each of these calcium binding sites, then what's going to happen is the conformation of this molecule is going to change. So let me draw its new structure down here. So basically what you get is you get the 2 lobes moving out from each other. So remember I drew it sort of hunched over up here, like so. Well, basically it's going to sort of spring back out. The 2 lobes are going to move out from one another. Okay, and also this linker between the 2 lobes, this was initially a linear polypeptide. It was amino acid followed by an amino acid, but it was a straight line of amino acids polymerized together. It's going to adopt a different structure now. It's going to adopt a secondary protein structure, which is an alpha helical structure. So it's going to adopt an alpha helical structure like so. Okay, so what you're going to get is these 2 lobes, now both with calcium bound to them. So I'll draw the calcium bound. So we'll have calcium in here, calcium in here, calcium in here, and calcium in here. And you've now got this alpha helical linker between them. So this is an alpha helix here. Alpha helix. Okay, and this structure now is known as a calcium calmodulin complex, which is often abbreviated, again using this abbreviation for calmodulin, it's often abbreviated to Ca2 plus CAM, so Ca2 plus Ca capital M. Right, okay, so when calcium goes up, because it's come in through these N and PQ type voltage gated calcium channels, calcium is going to bind to calmodulin to form these calcium calmodulin complexes. And these calcium calmodulin complexes now are going to activate the calcium calmodulin dependent kinase enzyme. But we'll discuss that in the next video.